Hi, thanks Ken, having me here. So the topic today are the flaviviruses. So as far as classification goes, they are part of the family Flaviviridae, which are all characterized by positive sense RNA genomes of 10 to 11 thousand bases. They are enveloped and then fall within a couple different genera within the family. Most of what I'm going to talk about, actually down here, is the genus Flavivirus, which are all, or primarily, arthropod-borne viruses. The mosquito-borne ones are probably the most well-known. Those would include yellow fever, West Nile, dengue, Japanese encephalitis, Zika virus. There are also a set of viruses which have very similar biology, at least in the host cell molecular aspects of it, that are carried by ticks. Within this country, these include some rare diseases like Powassan virus and deer tick virus. There's some more severe forms that are found within in Europe and sort of eastern Russia, Russian spring-summer encephalitis virus, tick-borne encephalitis virus, these sorts of things. Also of note, from the standpoint of human health, are the Hepaciviruses, a different genus which is mostly just characterized by hepatitis C viruses, and I will talk a little bit about Hep C. These are similar in terms of the genomic architecture, but as we'll see, there are some subtle differences, and I probably won't really touch on the pestiviruses at all, although these are a set of fairly important veterinary diseases, mostly found in the ones that are known are mostly livestock, and they tend to be, at least in terms of the genomic organization, they look a little more like HCV than the vector-borne ones. So the general outline of the talk will be some early discovery and disease associated with the viruses. I'm going to focus on yellow fever and dengue viruses. In the past, I've tried to cover other interesting flaviviruses such as West Nile and Zika virus, and I inevitably run out of time at the end, so we'll just kind of try to do two today. So some of that, then viral structure, binding and entry, replication, assembly, host cell interaction, and some touching on innate immunity, and finally a few slides on vaccine strategies and antiviral drug development and how those are used. So generally any flavivirus lecture starts with yellow fever because this was the first one described, at least by the Western world. The word flavus is Latin for yellow, so that's where we get the name from. This slide is just an excerpt from a medical doctor's notes from an outbreak in late 18th century Philadelphia, noting that I like that if you can read they made all the S's F's back then. I never figured out why they do that, but there we are. But the disease in this case was characterized by, most notably, the black vomiting, generally, which he notes is generally a mortal symptom, general jaundice, yellowness that appears in the patients, and he also noted, which is still true today in severe yellow fever cases, that there's often sort of a release of the fever later, so a few days after the onset, although then the patient can descend into shock later, which also leads to death. So the disease itself was fairly well described, but the agent and the transmission and strategies of prevention would have to wait probably close to 100 more years. So, as I said, it was well described and often appeared, or has been described to appear throughout the 18th, 19th century. The slave trade being a major cause of transmission or other movement between the tropical and subtropical areas of the Caribbean and South Central America, and then into the what is now the U.S. Characterized in its worst form by organ failure, acute hepatitis, characterized by the jaundice mentioned in the previous report. Early descriptions of yellow fever will always talk about miasma, bad air. These were the thoughts, but actually a Cuban scientist, Carlos Finley, is the first person known to have published the possibility that this is actually being transferred by mosquitoes. And this hypothesis was followed up on most notably later by Walter Reed, working as part of the U.S. military, and together with a researcher named Jesse Lazier, who is probably really more instrumental in demonstrating the mosquito hypothesis. And what Lazier did was let mosquitoes feed on people who were suffering from yellow fever, and then let them feed again on uninfected volunteers, generally military personnel. But what he found was that later on that this didn't work if you had the mosquito feed right away, but that there actually needed to be an incubation period in the mosquito. And he actually demonstrated this on himself and then died from yellow fever. So, but he probably got a nice paper out of it. Anyway, so that was demonstrated fairly conclusively by those experiments. There was also a lot of sort of hardcore stuff with more volunteers, where they have people sleep in the beds of somebody who just died of yellow fever, but prevent mosquitoes from coming in and show that those people don't get sick. So the epidemiology is actually quite impressive. Not something we would do today regarding the human challenge models, but figuring this out went a long way toward the discovery of how to prevent this, at least before we get to the era of vaccines. They further demonstrated that this was a filterable viral agent. Viruses had been described a bit earlier in the century. Let's see. So then further experiments demonstrated that there is, without the mosquito, there is no transmission, and it can be passed from human to mosquito during the first 3 days of infection, but then from the mosquito back to the human after a longer incubation period, shown generally here, that the mosquito acquires from the viremic human. There is a period, the extrinsic incubation period in the mosquito, and then the transmission to the naive human, and the cycle continues. Shown here actually for yellow fever and also for the dengue viruses, there is what's called the urban cycle, generally involves mosquitoes and humans, and the mosquito vector here, Aedes aegypti, is very well adapted to living with humans. It's found in any little bits of standing water that happen to be sitting around human habitation, small vessels, old tires, anything like that. It can be a mosquito breeding ground, so that once you have introduction into the urban setting, the virus can persist for quite a while. It's generally believed that most of these viruses have some sort of animal reservoir in non-urban or sylvatic or jungle settings. Often for yellow fever and dengue virus, probably some non-human primate, which is also cycled through a probably different species of but still the same genus, Aedes mosquitoes. And then sometimes there's some overlap with human encroaching or some interaction between humans in this cycle, which can lead to transmission to the urban cycle. One quick note about West Nile virus, which is similar, although it goes through birds, and the mosquito is the genus Culex. But so in the case of West Nile and several other flaviviruses, even ones that cause human disease, the human isn't always a viable host for transmission of the virus. So in this case, humans and also horses, which are susceptible to West Nile virus, are considered dead-end hosts. So the mosquito might bite them, it might transmit the virus, and that can lead to viral-associated disease. In the case of West Nile, that's generally encephalitis. However, the virus is not really adapted to growth in mammalian species, and the viremia experienced by the mammalian host is actually too low to efficiently transfer it back to the mosquito. So in this case, there's no actual cycle with the human, just you can get the virus to the human, but it ends there. Back to yellow fever, just to note that despite the vaccine, which I will get to at the end of the talk, yellow fever is still a problem in much of the tropical and subtropical world. Outbreaks continue to show up in regions of South America and also equatorial and sub-Saharan Africa. A major part of control of the spread of not only yellow fever, but also the dengue viruses and Zika virus, things that are carried by the Aedes mosquitoes, is control of the vector. You can see that the range of the Aedes aegypti, although prevalent throughout here in the Western Hemisphere in the 30s, mosquito control programs were effective at limiting its range back in the 70s. Although a lot of this involved insecticides, which were also somewhat environmentally unfriendly, and their use fell out of favor, which is not necessarily a bad thing, but from the standpoint of vector control, it has been resulted in sort of the reemergence of the vector and associated diseases in a lot of this area of the world. So novel methods of vector control are currently of interest and are being tested in a lot of these areas, including not only probably to a lesser extent chemical means, and also things like introducing sterile mosquitoes into the population in the hopes of preventing their breeding and reducing the populations. So the only other virus I want to go into much detail about historically are the dengue viruses, and I just want to mention these because as far as the mosquito-borne viruses go, this one is probably the biggest problem and the most prevalent throughout the world. So the dengue virus is actually the dengue viruses because it's defined by 4 serotypes, which some people will consider to actually be 4 viruses that result in similar disease characteristics. The serotype, the word comes from that they're defined as antigenically different. So if somebody is infected with one virus eventually, and you look at the antibodies within, circulating in their system, it'll recognize, the same serotype, but you can find other dengue isolates that that are less reactive or cannot be blocked by the antibody response to one. Infection with one serotype does probably provide lifetime or long-term immunity to that same serotype. However, in the short term, that individual appears to have antibodies that will neutralize all 4 serotypes, which wanes after about 9 months to become the serotype-specific immunity. This can get confusing. Is everybody with me on that?
the most part.
All right, okay. So all the different serotypes can cause severe and sometimes fatal disease, although within these serotypes there are further genetic differences, and there are some isolates that are associated with mild symptoms and some isolates that are associated with more severe symptoms, and these viruses are constantly kind of emerging and re-emerging within the countries where the virus is endemic. So dengue-associated disease currently is classified as dengue without warning signs, which is high fever, severe muscle aches, headache. It's generally fairly nasty, but there are other signs, severe abdominal pain, evidence of hemorrhage, bleeding gums, low platelet counts, which are indicative of more severe dengue, and then severe dengue is actually characterized by hemorrhagic symptoms within the individual that could lead to shock, and if the patient is not medically supported, can lead to death. Earlier descriptions of this you might see described dengue fever, dengue hemorrhagic fever, dengue shock syndrome. Some of the reason for the World Health Organization to sort of reclassify those as the dengue without warning signs is really about the question of how to care for patients who present with dengue-like symptoms. So there are no specific antiviral treatments. So treatment is generally supportive, fluid management, this sort of thing, to prevent or maintain people who have gone into shock or prevent that. So a big part of care for dengue patients is to determine who's in danger of going toward more severe dengue and who can be sent home to just recover from dengue fever, which will be, as I said, unpleasant, but probably is a self-limiting infection. So the various indications that are checked are used to sort of classify individuals and determine whether or not they need to be hospitalized. So the dengue viruses are present throughout, through the equator, and then, around the tropics of Cancer and Capricorn, through all the regions of the globe, Africa, the Americas, and Southeast Asia. Generally, they say about 2.5 billion people, that is 40% of the world's population, live within these areas and are at risk for dengue infection. Because this number is so high, this is one that often shows up in the introductory sentences of grant applications involving dengue virus. But this does lead to an estimated about just under 400 million infections per year, of which about 100 million are symptomatic. That is dengue, uncomplicated dengue fever or something worse. And in fact, about 500,000 of those are severe, requiring hospitalization, and some small percentage of those are lethal. So one complicating factor about management and prevention of dengue is that infection with one serotype is believed to give, well, as I said, it's been shown to give long-lasting immunity to that same serotype. A lot of this data was actually, again, done in human challenge trials during the years of World War II. A lot of it carried out by Albert Sabin, who was also famous for the oral polio vaccine. And he observed that, as I said, there's initial pan-serotype immunity, meaning the initial immunity seems to work against all the dengue viruses, that wanes to this serotype-specific immunity. But then when you have the serotype-specific immunity, infection with a different serotype is often associated with more severe disease than the first infection before seeing any of the dengue serotypes. And the theory that is generally accepted to explain this now, not entirely, but most people believe that at least some aspect of this is caused by antibody-dependent enhancement, which I'll go into a little more later, but basically, so if you have, these are antigenically different viruses, but they are related. So if you have antibodies that recognize one of these viruses and are able to bind to it strongly enough to prevent infection, for the other serotypes, they may bind but not neutralize. So what you have are viruses that are partially decorated with antibodies, but they're not completely inactivated or blocked from entering the cell. And because these viruses are attached to other antibodies, there's the potential that cells that have what they call Fc receptors, which bind to antibodies, are able to see these viruses and the antibody complexes, actually take those up, but then that results of an infection of those cells. So the presence of the less than effective antibody actually results in greater numbers of cells infected within the individual. So one of the, besides the conclusion that then this is maybe why the second or third or fourth dengue infections are going to be worse than the first one, this also causes many problems for vaccination, because if you're going to vaccinate against the dengue viruses, you have to be able to create antibodies that are good against all four serotypes, or else you might potentiate the possibility of the vaccine actually making infection with some serotypes worse. And a brief move away from the flavivirus genus to the Hepaccivirus genus. So hepatitis C virus, originally just characterized as a viral hepatitis that was not hepatitis A or hepatitis B, which had already been described, nor is it related to these viruses. Generally, this is a blood-borne pathogen. Infection generally gives you a mild acute form, but in most cases, people who are infected with HCV will go on to develop a chronic infection, so the virus is never cleared. And so it lives in the hepatocytes of the liver, and in 10 to 20% of patients, they go on to develop liver cirrhosis over a long period of time, which can also lead to liver failure, liver cancer, getting to the extent where the only possible cure would be a liver transplant. It was identified as a viral agent. People could pass it to chimps, which was the only viable animal model, and that's no longer in use. And then in 1989 is the first example of a virus being completely identified by only molecular biological means. It was cloned from a cDNA library of an infected patient using immune sera to recognize sort of viral-specific proteins. So they were able to get a certain amount of genomic sequence of the virus. I can define this. We don't need to go into detail, but I can answer questions about it later. Anyway, so scientists were able to identify a chunk of sequence from this that expressed some proteins that are recognized by HCV antiserum, which allowed them to then extend from that sequence to the whole genome. That allowed screening of blood products, which had been a major source of infection. So HCV has largely been cleared out of the blood supply. Still, new infections occur in this country and throughout the world, a lot of it due to intravenous drug use. So that there are still tens of thousands of new cases annually in the US, and an estimated 3.5 million chronic people living with chronic HCV in the United States. Okay, so now we're moving to the actual life cycles of the virus. We'll come back to this slide a few times just to give you a basic idea of what the flaviviruses need to do to replicate themselves. So there's entry up here. There have been several different receptors suggested for various flaviviruses. Probably a single definitive receptor is still not known for any of these viruses, although different proteins have been shown to be able to mediate viral entry into some cell types. So following entry, the envelope virus is internalized by clathrin-dependent endocytosis, where it gets into an endosomal compartment. The endosomal compartment matures and that results in lowering of the pH within the late endosome. That lowering of the pH causes the viral envelope proteins to change conformation, making them capable of fusing with the membrane. So once you fuse with the endosomal membrane, you can release the capsid and the nucleic acid. So the nucleic acid is a positive sense RNA, right? So that'll function as a messenger RNA, which can be directly translated by the cell machinery. So translation occurs sort of in close association with the endoplasmic reticulum. So some of the viral parts of this viral protein are expressed, inside the ER. Some of them kind of sitting on the cytoplasmic side of the endoplasmic reticulum. This single polyprotein is cleaved by viral and host cell proteases into 10 functional protein subunits. One of those subunits is an RNA-dependent RNA polymerase, which can then use the genomic RNA as a template to make a negative sense copy, and then it uses that negative sense copy to make more of the positive sense RNA, which can be used for either more protein expression or packaging into virions. So the virions are also assembled in close proximity to the endoplasmic reticulum. They actually bud into the ER and move through the cell's secretory pathway. At the sort of near the outside of the cell, you get to the trans-Golgi network, which contains a cellular protein enzyme called furin, which cleaves the PRM, which is pre-membrane protein of the flaviviruses, to the mature form M. So, and that results in the virus going from this somewhat spiky-looking thing to this smooth structure here. And then finally, moving through vesicular trafficking to the outside of the cell, you have budding and release of the viruses. And next, so this is the structure of the flavivirus.
So this has an envelope on it. How is the envelope? So the envelope is fusing with the endosome, and then the capsid inside is
released into the cytoplasm.
Does that capsid then uncode itself in the cytoplasm?
That's actually a good question. It's not entirely clear what the structure of the capsid is. So what I'm showing you here, that's the envelope. So the capsid, the flaviviruses don't seem to have a very organized capsid structure encompassing the RNA. It might be more of a sort of the RNA wrapped around several capsid proteins. The capsids do form multimers, so there is some structure going on, but whether or not there's a defined uncoding process or if the capsid and RNA are released in a way that allows direct access of the ribosome to the RNA, I think that question is still somewhat unclear. All I can tell you is that the sort of trying to analyze the structure inside the envelope by various means, NMR or things like that, have just given some sort of no particular organized structure that people have been able to point at. So, as I said, the classic structure of the flavivirus is actually the mature form of the envelope. So each envelope protein Each color here is a dimer of the envelope protein, so there's one here and then one next to it in a head-to-tail orientation. And so you have 90 of these dimers, or 180 total envelope monomers that form that coat the envelope. So you have this very regular structure, which, if you like doing this kind of thing, forms a T=3 symmetry. There's just another look at the envelope. So one here, head to tail, and one here. That's the dimer. So just another sort of description of the life cycle of the flaviviruses. If we just focus on the entry portion, the general strategy for entry here is receptor-mediated endocytosis. So again, depending on the virus, some type of protein receptor, there are likely interactions, charge-based, with other host cell proteins on the cell surface, maybe some non-specific ones with heparin sulfates, because in some cell types excess heparin can prevent binding. And then something that triggers the endosomal fusion, so movement through the endosomal process to get to the low pH, triggering the fusion reaction. Shown over here, on the far side, is the model for antibody-dependent enhancement of dengue viruses, or you can actually make this happen in vitro with any of the flaviviruses, given if you get the antibody concentration right, which is so binding of the antibody to the virus, and then the antibody binds to the Fc receptor, which then triggers endocytosis of the whole complex, receptor, antibody, and virus, and then that moves along to get to the same part where you get fusion of the virus with the endosomal membrane and release of the nucleic acid into the cytoplasm. So this is a type 2 membrane fusion protein. I don't know if you've done influenza yet. Okay, so that's similar idea, but the structure is somewhat different. The influenza protein eventually becomes this sort of big alpha helical structure that unfolds. These are largely beta sheets. The low pH environment causes these dimers to reform into a trimer, which then kind of forms this ice cream cone shape here. So this is a trimer of 3 envelope proteins. Down at the bottom here is the fusion peptide. So this is a hydrophobic domain which inserts into the target membrane, shown here. So you have the low pH change to the trimer, the fusion peptide inserts into the target domain, the envelope transmembrane domain. This would be the membrane of the virus itself. Then once this happens, the energetically favorable conditions cause sort of this pinching or bringing the membranes together, resulting in the formation of the pore through which the nucleic acid and capsid proteins will move. Shown here, a lot of this, just ignore these various things, but a lot of this was figured out through very careful mapping with different antibodies, which were able to show the different interactions and requirements for all of these processes. And then going back one more to the ADE model, one of the things that helped solidify this idea that this is actually happening in human dengue patients was that it was recapitulated in a mouse model of dengue infection. So there are a couple different caveats here, which I'm not going to go into. But in this model, these mice are susceptible to a strain of dengue 2, which if you infect them, results in death of the mice. And then if you give them dengue 2 antisera, you can, about half of them will survive. But giving that same antisera to mice infected with, giving dengue antisera elicited against dengue 1 or dengue 3 or dengue 4 to those same mice that were infected with the dengue 2 strain results in more rapid decline of the mice. And this was also confirmed to show that that the dengue 2 immune sera binds to dengue 2, not surprisingly, more strongly than these other antisera, and that it's able to neutralize in the case of dengue 2, but not in the case of these other antisera, suggesting that you can recapitulate a lot of this secondary infection with a heterologous phenotype being more severe in the context of an immune response to a different serotype. So that's entry. So consider ourselves inside the cell. So the next steps for the virus are translation of the proteins, replication, and egress. So this is the flavivirus genome organization, so single positive sense RNA with, it has a 5' cap and sort of fairly extensive 5' and 3' structures. A lot of these are required for different aspects of replication, and in fact there's interaction between the 3' and 5' untranslated regions, so there are regions here that are called the cyclization sequences, which have to be complementary. So that's some part of the replication process. The protein, as I said, is translated as a single polyprotein, which is cleaved as it's being translated into these 10 subunits, the structural proteins, the capsid, the premembrane membrane protein, the envelope protein, and then NS or non-structural proteins, NS1, 2A, 2B, 3, 4A, 4B, and NS5. Shown here, sort of the topology of the thing. So this is the cytoplasmic side, and this is the envelope, the reticulum of the ER. The ER inside here, lumen is the word I'm looking for. You see that there are signal sequences within the proteins that result in translation of the capsid in the cytoplasm, then movement to the ER, and translation of the envelope or PRM and envelope proteins in the ER, and then a lot of this going back and forth throughout the translation process. Notably, the NS2B and 3 proteins form a protease, which is required for several of these cleavages, shown here, the arrows coming from 3 and 4. These little scissor diagrams are where host cell proteases are required for cutting, cleaving these proteins. And then the NS5 protein includes a polymerase, which is required for making the RNA, for copying the RNA, making the negative and positive sense progeny. And also it has a methyltransferase domain, which is responsible for capping the RNA, as well as it has functions in immune evasion. The hepatitis C viruses are of similar architecture, but a little bit different. These are not capped. They actually have an iris, similar to the picornaviruses. They have, instead of PRM and E1 and E2 proteins, and the NS5 protein is divided into NS5A and 5B. 5B is the the RNA-dependent RNA polymerase. NS3 together with NS4A is the viral protease, but generally this sort of idea of the translation in and out of the membrane in close association with the ER membranes holds throughout all these viruses. So within the cell, this is work from a lab in Germany, that of Ralph Bartenschlager, had done quite a lot of impressive electron microscopy, looking at the structures, membrane structures formed in flavivirus infected cells. It had long been known through other people's EM work that in the flavivirus infected cell, the membranes form all these kind of crazy shapes that are described depending on how good your resolution is as these convoluted membranes or vesicular packets. So what was done here is actually cryo-electron microscopy, sectioning through frozen sections of infected cells and then sort of reconstituting the 3D structures of what these all look like, which led to this model that, if you look down here, that the proteins are translated into these, these invaginations within the ER, where on the inside, which is still topologically part of the cytoplasm, is where you have the RNA being translated into the ER. If we look here, we get this a little bit more closely. So you have this invagination into the ER. Here are the RNAs being translated and then replicated by the polymerase and the associated proteins, either in or in the cytoplasmic or ER lumen, depending on how they were translated. So, let me just move back one. And then it's believed that you have these little sort of exit sites, which are in close approximation to ER membranes. They're shown here in these white arrows, so that it's believed the capsid and associated positive strand nucleic acid then moves into the ER here, sort of in the ER membranes across from the exit site, which is where it then acquires the envelope by budding into the ER, so that formation of the virus happens in the endoplasmic reticulum across from where the protein was, where the RNA was replicated. So all of this is very spatially regulated as well as enzymatically regulated. And great. Okay, so also within, while all this is going on, the cell is basically trying to fight back. So before we get the adaptive immune response, antibodies, T cells, that sort of thing, there's quite a lot of innate immune responses to viral infection, which serve to tamp down viral infections that are constantly being sort of countered by the virus. So there's this whole cat and mouse game going on between the infected cell and the virus. So for the RNA viruses, a big part of the antiviral response involves first recognizing that the cell is infected, and this is often done by the presence of double-stranded RNA, which is then because you have this negative sense intermediate in terms of replication, is sort of a byproduct of viral replication. So there's several proteins called RIG-I, or MDA5 is another one, that recognize double-stranded RNA. And when they do, they undergo a conformational change that allows them to signal to various components of the innate immune system, resulting in, do I have that on this slide? If we move all the way down here, phosphorylation of a protein called IRF3 and activation of proteins called NF-kappa B of the NF-kappa B family. And that results in production of interferon, which is sort of the major mediator of the antiviral response, and then that feeds back onto the cell. So interferon is secreted, it signals to neighboring and, in fact, the infected cells, and that results in production of a variety, some hundreds of different proteins, which have various antiviral effects. Notably, like some of these proteins are targeted by the viral proteins so that the innate immune response is dampened down. For example, this protein STING is another sensor of infection that is a target for the NS2B3 protease of most of the flaviviruses, the mosquito-borne ones anyway. Another aspect of this is blocking interferon signaling itself. So when interferon binds to its receptor, it results in phosphorylation of these associated kinases, the JAK kinases, which then phosphorylate these proteins, STAT1 and 2, and then that complex goes and turns on most of those interferon-stimulated genes that I had just mentioned. So the STAT2 protein, if I'm seeing it right, is a target for all the different flavivirus NS5 proteins. So these proteins can bind to STAT2 and result in their degradation. Interestingly, they have slightly different mechanisms of doing this, but most of the ones that have been looked at do have this phenotype of being able to degrade the STAT2 protein and block interferon signaling. So this is crucial for the virus to be able to replicate itself within the infected host. One thing that's notable is that several of these flaviviruses, like the dengue viruses or yellow fever or Zika virus, are unable to do this in murine cells, so they're not adapted to grow in mice. So people have found that if you infect mice, wild-type mice with these viruses, the mice don't get particularly sick. However, if you knock out the interferon response, or in fact just STAT2, you can make a model in which the mice, where you can see virus replication and there's a pathogenic effect on the mice. West Nile virus is able to block mouse STAT2, and that grows just fine and is in fact lethal in wild-type mice. Besides, I mean, these are probably some of the best studied examples, but there is a vast array of interactions between the different viral proteins and different host proteins, which are antiviral, that are required, that occur during the infection of the host cell. And although the virus is able to overcome a lot of these effects and replicate itself, you should also be aware that if the host cell if there was no interferon or innate antiviral response at all, the virus would be all of these things would be rapidly lethal, which is not the case. So there's clearly still this balance being struck in the current, race for supremacy between virus and host. So I guess we're doing about right. So I want to sort of finish up with some of the preventive or therapeutic ways of addressing all of these viruses. So vaccines are generally a good one for preventing viral infection. The yellow fever vaccine is one of the earliest and most successful vaccines that have been discovered. So in 1937, Taylor and Smith took a pathogenic strain of yellow fever and passed it multiple times, first through cultured, basically ground up mouse embryos, and then through chicken embryos, and then finally through chicken embryos in which the brain and spinal cord had been removed. This being because some of the pathogenesis of yellow fever, at least in the mouse model, involved infection of brain cells and neurons and encephalitis. So after this multiple passages, or going through this, they continually tested these viruses in mice and in non-human primates to look to see if these viruses were attenuated, and eventually got to a strain they call 17D, which seemed to be attenuated. It had a loss of viscerotropism, that is growth in the body, which was measured by a lack of viremia. It had reduced neurotropism, so if it was injected into a mouse brain, it was no longer, or at least less lethal than it had been. And it was no longer competent for growth in mosquitoes, also very important if you want to block transmission of this thing. The current vaccines, 17D doesn't really exist anymore, but there are 3 derivatives of this which are propagated from seed stocks that are currently in use. If you look at these 3 different ones, and then look for what's in common between them and the pathogenic strain, there's something like 20 amino acid differences and 4 nucleotide changes in the 3' untranslated region of the vaccine strains compared to the wild type one. The actual mechanism of attenuation, even after all this time, is still somewhat unknown. So we don't know exactly which of these differences, or it's probably a combination of them, but we don't know exactly which ones are responsible for making this less lethal, but allowing it to remain antigenic. A much more sort of targeted approach has been taken to dengue vaccine development. So there is currently one that has been licensed in several countries, and this actually takes advantage of that attenuated yellow fever vector. And so the molecular clone of the yellow fever vaccine strain was synthesized, and then the envelope proteins of yellow fever were taken out and replaced by dengue 1, 2, 3, or 4 PRM regions, PRM and envelope regions, so that when you infect with this, the host will make an antibody response primarily to the envelope proteins, which are of the dengue proteins. And that works. It is attenuated. It can produce an antibody response to the host. And it had gone through large clinical trials, and then is currently in use in, as I said, some, well, it's on the next slide. I think it's about 13 different countries. But other strategies that are currently in use are molecular deletions of parts of the 3' UTR, these Delta 30 vaccines, which were developed by the National Institutes of Health, are also somewhat attenuated, or at least some of the strains are attenuated. The dengue 2 was actually a little bit too hot even with this deletion, so that's within their sort of tetravalent, all 4 serotype group. They have attenuated versions of 1, 3, and 4, and then a 4 with the dengue 2 envelope stands in there. And then there's a couple other attenuation strategies. So Takeda has one where I think they had a attenuated dengue 2 that was done by serial passage, and then they put the different envelope proteins of the other serotypes into this backbone. So the first one, which is the Sanofi Pasteur approach, which they call Dengvaxia, as I said, the backbone of the yellow fever virus with the different dengue, PRM, and E. So you do get neutralizing antibodies in the vaccinees. The clinical trials looked like it was effective, although not great, but they would see something like 50 to 60% efficacy with some difference in the different serotypes where some were blocked better than others. It is in use in both Latin America and Southeast Asia. 13 countries license it for use in kids who are over 9 years old. And one of the problems with this is that in younger vaccinees, under 9 years, they've actually observed a slight increase in hospitalization due to the vaccine, which further analysis suggested that in naive individuals, so people who have never seen any dengue strain, this vaccine may actually increase the risk of severe dengue, possibly again, if the antibodies it's producing are not potent enough that you have this ADE effect coming into play. And this, of course, is now being litigated in courts throughout the world, although it is still in use in quite a few countries. And then antivirals, as I said, there are none currently for the mosquito-borne flaviviruses, but there are for hepatitis C virus. So early on in the history of HCV therapy, the standard of treatment was interferon plus inhibitor of viral replication called ribavirin. It was sometimes effective, giving what they call the sustained viral response, i.e. like no virus is eliminated from the body. But this was a long treatment lasting months with very many nasty side effects. And the pharmaceutical industry had been very interested in direct-acting antivirals, initially targeting protease inhibitors, although these had the disadvantage of quickly developing resistance in the viral strains. And they also required extensive dosing and had some nasty side effects. And then Gilead developed or bought a company who had developed an inhibitor for NS5B. So this is a nucleoside analog that is recognized by the viral polymerase. It's there, and it's incorporated into the RNA chain, and it's unable to, the polymerase is then unable to continually add to it, so it's an RNA chain terminator. This is initially called sofosbuvir, which they renamed Solvati. I don't know how they come up with these names, but there's got to be somebody who's paid a lot of money to do this for some reason. But anyway, oops. I lost my last slide. Sorry. Anyway, so that was very effective. And most recently, Gilead has also developed a drug which they call Harvoni, which is an inhibitor of the NS5A protein of hepatitis C. And that is currently their current therapy is a combination of these two drugs. So you're blocking the polymerase, blocking RNA replication, and blocking something that NS5A does. That actual mechanism is unknown, but it's known that if you were to in vitro try to find resistant mutants to the dipisphere, that they map to NS5A. That's why it's believed to be an NS5A inhibitor. So these drugs are very effective to the extent where that hepatitis C is, it's actually not viewed by the drug industry as a major problem because it's viewed as solved, although there's still many people who require this therapy, and one of the problems is it's ridiculously expensive. Currently about $84,000 for the full treatment in the United States. But it does work, so I'll just, leave you to judge what the best way, how the drug industry comes out in this one. They solve the problem, but they're charging people a lot of money to live. So that was my last slide. How am I doing on time? All right. Yeah, I finally got this right after 3 years.
You got more questions? I will post the PowerPoint and then the audio later today, so this will be available.
Yeah.
Is the antibody enhancement you mentioned known to work across other flaviviruses? I thought I heard something with Zika, like if you got it.
Yeah, so it's been, it's unclear. So in that, I think this is maybe an example of maybe asking the wrong, or using the wrong model. So people have shown that you can enhance Zika virus infection with anti-dengue antibodies in the mouse model. But no, there's no evidence of this in humans. So looking at, say, during the outbreak, the women in Brazil who had, Zika was associated with birth defects, there was no correlation between dengue seropositivity and the outcomes of pregnancy there. And it's also been modeled in non-human primates, so they're infected with dengue and you wait a year and infect them with Zika, and there's no difference in replication of Zika in those models in those animals. So it's still sort of up in the air. You can kind of experimentally create conditions, but you can do this almost for the ADE. You can do this almost in any case if you even if you have an antibody which is potently neutralizing, if you give a little bit of it, so there's not enough of it, you can get some enhancement kind of phenotype. So you need to know what your modeling actually occurs in real life. There is some evidence now that it might go the other way, that in the macaque, and I don't know if we see any human studies yet, but that Zika immunity may be potentiating for dengue infection, which we might see this in the future, given that there's now a large number of Zika immune people in South and Central America. So yeah, I guess that's the long answer to your question is maybe, but in people, probably not so much.
Let's thank Alec.